to present something about the B site. First of all, I want to mention, we are in the division CEDA and the Office of Business Informatics. So our office is mainly manage all the master data program in the CEDA, and we provide the data service to the reviewer and the inspect in the CEDA. So during the routine operation, and we recognize a lot of challenge when we capture the B site, because we are not only capture B site, we capture a lot of data from incoming submission, including the products and the facility, B site. That's a lot of information. So first of all, the reason we want to capture B site is because due to the short term of GDUFA due date, it's about 10 months or 15 months. And special inspection takes a longer time. That's why we want to get information on the B site as early as possible. We can transfer all the information to the inspection team. They can arrange the resource and time to get this done. So first of all, we go back to look at current guidance. Basically, we have eCTD conformance guidance, and in the guidance, it's very clear states. So all the NDA bioequivalence summary table should be provided in the section in the module 2.71, table 10. This is eCTD and conformance guidance. So if you look at this is a URL, you can look at the real PDF file. Based on the SOP we have, so ideally, an applicant should submit all the B site information, at least the table 10 in the module 2.7. For us, the most easiest way just to go to the module 2.7 for the table 10. And in the table 10, you will see the study title, study number, and what is VIVO study or VITRO study, what the site is, the address, the principal investigator. All the information should be there very clear for us to capture. So this is the real process we have today, because sometimes we are not able to capture information in the module 2.7. Then we have to go to the second place and even go to third place. The worst case, sometimes we have to go to from module 1, module 2, module 3, module 4, module 5. Try to allocate all this B site information, because this information is very critical for us. The first challenge we have is sometimes when we look at module 2.7, there are multiple tables, sometimes a lot of tables. And for us, we always look at table 10 first, because that's the table recites all the key information of the B site. And if you look at this example, sometimes people provide a table, but the table number just mislabeled. They use table 6 in this example, table 6. Sometimes they label as a table 11 or 12. We have to go through all the table, really look at the table named as study information, because we want the information in this table. Second example we have is also kind of challenging. A lot of case, we have a VIVO waiver because we don't need a VIVO. But still, you may have a VITRO study, right? You still have a VITRO study B site, may need to be inspected. In this case, there's no information in the module 2.7.1. And in this example, actually, the information resides in the module 1. In the module 1, there's no table available. It only have a study report. The usual study report contains 50, 100 pages. And in this case, it's difficult for us to really define what exact B site is, the location, the address, and all this kind of detailed information. And The challenge we have, even sometimes we successfully allocate this information. When you look at detail, it's still missing a key information. I mean, the key information is, for this example, we know the B study was performed by ABC Pharmaceutical. You look around all the information, there's no address list where the ABC Pharmaceutical is. And then it will be difficult for our OPQ team, if they want to perform inspection. It's hard to allocate where the ABC Pharmaceutical is, especially if they have a multiple location, right? Maybe some in domestic location, some in overseas. So this is something we always see from incoming submission. And for this example, the reason we want to capture the B site, this detailed information in the front, because we really want to reduce the processing time. The more early, the more accurate information we can provide to the downstream inspector, more like the inspection can be done before the due date. That's very important. We don't want to go back, just before the due date, we recognize, okay, we went to a wrong place, right? So in this case, we have seen quite often, there's no information available. I mean, applicants put all the information into Module 5. In the Module 5, it's often we see you have 15, 10 different studies. Each study will present by study report. Each report have more than 50 pages. Then we have to dig in these 50 different study reports, try to allocate all this key information. That's a huge challenge for us. As a summary of a challenge, so as I say, B site information is the key, and the key components of the B site is the name and address. Because we, as our team, we don't perform a review detail, but we want to identify this key information, then provide the other team to do their job. And also B sites appear in different formats. We expect the table format, in table 10, the information would be very clear. And we don't spend too much time to dig in, identify, do a comparison. But unfortunately, we see a lot of different formats, table, study report, in text format. And also, this is the biggest key. The B site information is not consistent placed in a central location, which we expect. So I think potentially if we cannot identify B site, the inspection cannot be on time. Ideally, the review session will get delayed or something get postponed. And the central message I want to send to you today is we really expect all the B site information. I'm talking about, these key components: the name, study number, study type, principal investigate, and the address. So it should be allocated in the table 10, module 2.1. So this is a table 10 looks like. You have a study number in the front and the study type, and you can circle what the B study you want to present, the Vivo or VITRO. And then you have a clinical site, you have an address, you have principal investigate. So as soon as we have all this information in this table, we capture, and the downstream inspector, they can arrange. Because a lot of B site is not in USA, it's in overseas, in India, in some location. Really need the time to arrange. And this UI will give you, if you really want to see a PDF file format, you can click this link and bring you to the detailed document. And this is additional source. If you log in FDA.gov, you can see all the eCTD related documents about this BE site. Okay, this is for the B side. I hand over to the next topic to my colleague, Derek Smith, talking about the manufacturing facility. Thank you.
Thanks, JJ. So we'll switch gears a little bit here, and JJ was talking more about the data capture of the BE site information. I'll be talking more about the manufacturing facilities, commercial and exhibit batches within the application, and where our expectations are, what challenges we see. And so, going back to the master data, inaccurate manufacturing facility information. So in the form FDA 356H or Module 3, if we have inaccurate information, it really causes issues, because as they come in with the master, as JJ's group looks at it at the master data level, they try to compare and understand the information to make sure they have the right site to give to us on the assessment side to make sure that we're inspecting the right sites. And so, our recommendations are, use the identifying information that's most accurate for the current agency records. You can normally find that information in the last EIR, the Establishment Inspection Report from the facility. And if you do have changes, if you have a new building next door or a different address or things like that, make sure you're contacting Office of Regulatory Affairs to notify them of those changes so that they can get that information within their databases, so that then CEDR can then use that information as we're assessing the applications. One of the other common issues for us in the model that we use that is used to capture the data and then propagate to us to assess the facilities is extra facilities that aren't supposed to be on the 356H are being listed, and then it kind of bogs down the amount of facilities and things that we're looking at on the application assessment side. So recommendation there is to check out the form FDA 356H supplement. It gives pretty good instructions as to what should be on the form and what shouldn't. Field 27, that's going to be the manufacturing, packaging, and control sites for both drug substance and drug product. And any facilities that don't meet the FDA form 356H criteria based on the supplement guidance, we'd recommend that you put those in Module 3 in the facility listing there, so that all the information is available for us to evaluate. And so what do we commonly see within the application? So what we'll see is that the bioequivalence, bioavailability, and stability batch manufacturing, packaging, and testing sites, which are not proposed for commercial manufacturing, may not be listed very clearly within the application. More commonly with the sites that test the BE batches, and by test I mean the release and stability testing of those batches. And so, I think one of the problems that we see is the form FDA 356 really doesn't tell you what to do with those facilities. And the reason that we do need to know about them are the two CFR references I have here, 31494A9 and 31450D12B, requiring the manufacturing facility information for those batches be submitted if those sites are indeed not the commercial manufacturer. So recommended best practice, include those sites in Module 3, and within Module 3, make sure you notate that the facility is not proposed for commercial operations, but manufactured the exhibit batches. And I can say that I think I've given this presentation a couple times now, and we do see some sponsors of ANDAs really putting together nice tables within Module 3 in 3.1 that do give that information, that say, this is for exhibit batches, this is what the facility did. For commercial batches, this is what the facility is proposed to do or not proposed to do. One of the biggest pain points for us as we're reviewing the applications and really jeopardizes both meeting the GDUFA date in a timely manner, as well as perhaps extending your GDUFA date due to an omission of information. And so the drug master file manufacturing and testing facilities, the FDA 356 instructions does state include all. And I think what sometimes struggle with is, what do we mean by all? It's not just that final DMF manufacturing facility, right? Yes, that is the source of the API, but is that the site that's release testing the API? Is there a critical intermediate that's more upstream? Is that final site only doing a one-step synthesis? But really there's another site that does the major structural, the synthesis that gives you the major structure of the API. And then any sterilization, micronization sites as well. And so the other common thing we see with the DMFs are that some DMFs will list 3-4 manufacturing facilities that may supply the material. For us, that when you only list one in Module 3 or on the 356H, but the DMF has 4, we then have to question, are you using all 4? Are you using just one? And then there's going to have to be the back and forth with IRs, and so trying to get things moving as fast as possible to not delay inspections and not delay evaluation. What we recommend is if the DMF does have multiple sources and you're only using one source, then you specify that within the application so that we know that you're only using that one source and you're committing to only using material from that one source. One of the other things that we commonly recommend is if you're only going to use that one source, making sure that there's a statement that says you're only using the source. If you intend to use another source from the DMF, you will submit a supplement as appropriate to add those additional facilities to the application. Another recommendation is that within the LOA, that's going to go in the application is also going to be on file from the DMF for you. That LOA from the DMF order could specify that you are only using material from these facilities, and it helps us have more accurate information to start the assessment process as early as possible. And just some closing thoughts here. The goal is to get the information, assess the information, and be able to make timely decisions. Foreign inspections take up to 6 months to plan, so the faster we have that information and can process it, it's accurate, we can get that started. And so submitting accurate information in the appropriate sections really facilitates the efficient quality assessment. If we have to dig within the analytical methods section to find out that there was another testing site that was used that wasn't listed in Module 3, we're not going to find that until month 5, and there's only 5 months left in the clock. Considering impact of missing facilities on the goal dates. If we do find these facilities, we only have a certain amount of time. We might have to send you an IR. That IR then might cause the goal dates to shift to allow time to assess the facility. And then one of the other common things with ANDAs is changing facilities. So you submit your application. You have sites 1, 2, and 3. Site 1 is found to be not compliant with CGMPs, and it's going to hold up your application. It gets a warning letter, for instance. And what do I do now, right? Obviously, yes, I want to get the ANDA approved. I want to get to market as soon as possible. But what is the impact of that change if you're going to take Site 1 that's under a warning letter out of the application and not propose it for commercial, and go to a different site? So think about what that change is. What were the issues at the site with the warning letter? How is that going to impact the data in your application? And again, the reminder here is most often that site, if it's the drug product site that was used to manufacture the submission batches that are going to support the ANDA, it does still have to be listed in the application. You can't just take it out of the application, right? It has to be there per the regulations. And then the other thing to think about is there's a guidance, alternate sources of active pharmaceutical ingredients in pending ANDAs, and it kind of defines when there might be a need for additional BE studies based on changes in API source. And so I think that while it's very generalized and it's obviously a case-by-case analysis, it does kind of give you some guiding thoughts as to understanding the impact of changing your facilities and the scientific principles of understanding the quality of the material that was used to support the ANDA from those sites. Even though that guidance is for APIs, right? The same would apply to the DP site, right? So if there's a question of the quality of the drug product that was manufactured to support the BE studies or to support the submission, and you're changing sites, the agency is going to have to assess that impact and determine if additional information will be needed.